Alright, let's go ahead and continue on now with section 2 of chapter 5. We're going to look at building organisms through gene expression, the information DNA directs in production of molecules, and we're just going to continue on from what we were discussing last time. If you recall the overall flow of information, we of course go from transcription to translation. Transcription is reading the information encoded in the DNA. So if DNA is like a cookbook filled with recipes, transcription and translation are like cooking, of course. In cooking, you use information about how to make the chocolate cookies and how to actually produce them. In an organism, the information about putting together the proteins, a sequence of amino acids, is used to build the proteins the organism needs in order to function. So we're going to examine transcription, the first step of the two-step process by which DNA regulates the cell's activity in formation or synthesis of proteins. So looking at the overall transcription process, first we have to recognize and bind. So once RNA polymerase recognizes a promoter site, it binds to one strand of the DNA and begins to reading the gene message. So we have a promoter site. It's a site that actively engages and gets RNA polymerase to start this transcription process. And if you look in the diagram here, we're obviously moving in a direction. If you recall, DNA has they're anti-parallel. We have 5' to 3' and 3' to 5'. It's not really important that you remember that, but we transcribe as DNA strand is processed through the RNA polymerase. The RNA polymerase builds a single-strand copy of the gene called the messenger RNA transcript, which is a complement of one of the strands, the sense strand of the DNA. It terminates when the RNA polymerase encounters a code signaling the end of a gene. It stops and releases the messenger RNA transcript. So the termination site in messenger RNA transcript is shown here. The capping and editing. Before the messenger RNA transcript can be translated into a protein, a cap and tail are often added for protection, and to promote recognition, non-coding sections are removed. The messenger RNA transcript leaves the nucleus to be translated into a protein in the cytoplasm, or goes to the endoplasmic reticulum, the rough ER, or the shop floor, if you recall our analogy. Before any of this can happen, though, the RNA polymerase has to move down the strand. We have to unwind the helix in order to get to open and unzip that region of DNA. So I just wanted to point that out. We're unwinding the DNA so we can get the machinery in there to make the messenger RNA transcript during the transcription process. So transcription is the first step in the two-step process of which DNA directs synthesis. In transcription, a single copy of one specific gene within the DNA is made in the form of a molecule of messenger RNA, which moves where it can be translated into a protein. So translation is using information from DNA to bind usable molecules. So once messenger RNA molecule moves out of the cell's nucleus into the cytoplasm, the translation process begins. In translation, the information carried by the nucleotide sequence of the messenger RNA is read in groups of 3 as a codon, and ingredients are present in the cell's cytoplasm are used to produce a protein. The process of translation is like combining all the baking ingredients listed in our chocolate chip cookie recipe to produce the cookie. So there are several ingredients that must be present for in the cytoplasm for translation to occur. We have to have the 20 free amino acids, ribosomal units, there's 2 of them, and transfer RNA, which is the decoder ring. One part of the molecule transfer RNA has the amino acid, the other end of it has the anticodon, or that unique decode message for, or that 3 nucleotide sequence unique for that particular amino acid. So several ingredients to recap. First, there must be a large number of free amino acids. The amino acids are the raw materials for building proteins, an essential component of our diet. Second, there must be molecules called ribosomal subunits, the moving protein production factories where amino acids are linked together in the proper order to produce the protein specific or specified by the messenger RNA transcript of the gene. Finally, there must be molecules that can read and translate the messenger code from a base sequence into a protein. These molecules are called transfer RNAs and are interpreters of the messenger RNA code, linking specific bases in messenger RNA with specific amino acids. Because they play such a central role in translation, we examine them more closely. So here's the transfer RNA. There is the unique attachment site. It's that codon. The UAC is, you can see the purple, blue, and yellow. Attached to this transfer RNA, you can see the amino acid at the top, and that's the specific amino acid for that particular sequence. So this is the decoder. So this takes that three amino acid, the nucleotide code, and places that amino acid onto a growing chain or necklace. So here are the groupings of three on the messenger RNA, and then we get now the three on the codon, anticodon. This is what the attachment site are referred to as the anticodon. And that's how that works. So human translators can hear a word in one language and say those same word in a different language. Transfer RNA functions in a similar manner. Picture a two-sided molecule. One side of the amino acids attached. This amino acid can be any of the 20 different amino acids. In other words, there are separate transfer RNAs for each of the 20 amino acids. On the other side of the two-sided molecule is the attachment site consisting of a 3-base sequence that matches up with a 3-base sequence on the messenger RNA transcript. This matching up enables the tRNA molecule to attach to messenger RNA. Thus, the transfer RNA translates the code into the messenger RNA into an amino acid. Now, here's translation. Now, recognize that we have two subunits of the ribosome that clamp on to the messenger RNA, and so we're attaching. You notice the three binding sites. We have the early site, the mature site that actually recognizes the sequence, and then the amino acid is picked off and then is dumped off the chain. So the amino acid comes in. If it matches that particular sequence, and you can see that it requires a perfect matching, so you can see now the complement to each of these nucleotides are met, then the amino acid is now added to the growing peptide chain, and then the uncharged transfer RNA, or the one that's had the amino acid removed, is dumped out, sort of like a empty cartridge on when you expel a fired bullet. So the empty cassette is immediately expelled. So you can see it's moving forward, adding, and then dumping the uncharged. That goes back into, by the way, the cytoplasm to become charged again, and then it plays again into the role. So this goes on and on and on through the sequence until it gets to a stop sequence. And that particular transfer RNA that comes in, its function is to cause the ribosome to stop and to disassociate. And that stop, or that punctuation message, is then acted upon. So let's translate the following messenger RNA. We have CGA, GCA, UGG, GCU, UUAG, as it goes on. And using the chart that I gave you in class, but you can also look it up. I would provide you with the proper, without a translation or lookup table. Doing this would be impossible. So if you had that lookup table to find that a CGA represents methionine, GCA represents glycine, so that would mean that each of the methionine, glycine, phenylalanine, arginine, blah 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 goes on, would have these triplet codes complement so that the amino acids could be placed. So without code, this would be impossible for you to know this. And I just wanted to bring this up, that you have to have a lookup table, which would be provided if I were to ask you to do that. So translation occurs in 3 steps. First, recognize and initiate protein binding. Translation begins in the cell, the cytoplasm, where the ribosome, essentially 2-piece protein binding factory, recognizes and assembles around a start sequence, which is always AUG. That's the start, and each other on the messenger RNA transcript. So as the ribosome subunits assemble themselves into a ribosome, one side of the transfer RNA recognizes a start sequence and binds that messenger RNA at that point. That initiator transfer RNA has the amino acid methionine bound to its other side. This will be the first amino acid in the protein to be produced. So if we look here, the only one that has the first as a methionine would be number 1. So you would know right away, if you had the lookup table, which one was the methionine. So the UAG would fit there, right? It doesn't, does it? So the AUG, we have, where's the AUG? AUG, there is the start codon right there. So we have to look for that particular sequence for the start. Knowing that you did not know that, I'm not going to have you memorize, but this was a good illustrative process to be able to pick out where the start sequence is. Now, this is a high-paying job in terms of looking at sequences of DNA, and it's more of the intelligence in terms of going through and transferring all that information from the Human Genome Project, but I just wanted to point that out. Okay, so on the messenger RNA transcript, the ribosomal subunits assemble themselves into the ribosome, and the initiator has the amino acid methionine bound to its other side. This would be the first one, as we found. Step 2 is we elongate. After the messenger RNA start sequence, then the next 3 bases, or the codon of the messenger RNA, specify the amino acid carrying the transfer RNA molecule should bind that particular one in the next sequence. If the next 3 bases on that messenger RNA are GUU, for example, transfer RNA molecule that recognizes that sequence will attach. At that point, GUU-recognizing transfer recognizing molecule always has the same amino acid valine attached. So the ribosome then facilitates the connection of the second amino acid to the first. The process continues in the same manner. The next 3 bases on the messenger RNA specify the next amino acid, and so forth. This is the beginning of the protein synthesis and protein chains, like beads on a string. The messenger RNA continues to be threaded through the ribosome until the ribosome, moving down, reads and translates its message until in these 3 base chunks or codons. Each 3 base sequence specifies the next amino acid, lengthening and growing the amino acid chain or the strand. After the amino acid carried by the transfer RNA molecule is attached to the growing protein, the transfer molecule detaches from the messenger RNA and floats away. That's the uncharged one, and it becomes charged again in the cytoplasm to be used again in later processes. Step 3, we terminate. Eventually, the ribosome arrives at the 3 base sequence on the messenger RNA that signals the end of translation. Once the ribosome encounters this sequence, the assembly of the protein is complete. Translation ends in the amino acid strand, and messenger RNA molecules are released from the ribosome. When it is complete, the protein, such as insulin or a digestive enzyme, may be used within the cell or packaged for delivery via the bloodstream to somewhere else in the body where it is needed. Following the completion of the translation, the messenger RNA strand may remain in the cytoplasm to serve as the template for producing another molecule of the same protein. In bacteria, messenger RNA strand may last from a few seconds to more than an hour. In mammals, messenger RNA may last several days. Depending on how long it lasts, the same messenger RNA strand may be translated hundreds of times. Eventually, it is broken down by enzymes in the cytoplasm. So, to answer, shows the messenger RNA is transcribed from the DNA sequence below. So we have TTA, TCC, TTT, ACT, CAT, going 5' to 3'. So which answer shows the messenger RNA transcribed? So first thing we look for is make sure that we have uracil instead of thymine, and that the complement strand, which shows the messenger RNA transcribed from that 5' to 3', is that it would have to be the complement. So A's is what we would expect, AAU. So number 2 is the AAU, AGG. Also we have number 4 that's got the UUA. So we can't have thymine, so we have to be uracil. So AAU, AGG, AAA, UGA looks like number AUG, AGU. So actually it appears TTA, so we have to have AUG first. That's the beginning of the sequence, is it not? This actually shows messenger RNA transcript. So let's think about this. So I believe number 2 would be the correct one. 5' to 3', as it makes the yeah, so it's number 2. That's incorrect. It should be the answer should be number 2. The book company is wrong on that one. It would be AAU, AGG, and then on, because none of that would make sense if it were let's see, CAT, G, AUG would be AUG. Oh, they were sneaky on this one. What they did is they moved from 3' to 5', and that would be correct. So the 5', the 3' message would be AUG, starting TAC. Aren't they little sneaky ones? So AUG, so I was wrong. That would be a common mistake, by the way, a very quick one to point that out. So TAC is AUG, TCA would be AGU, TTT would be AAA, CCT would be GGA, and ATT would be UAA. Aren't they little sneaky ones? So number 1 is the correct answer. Number 2 would be one that you would select if you just wanted to be lazy and to pick that one like your instructor did right away. So number 1 would be the correct answer. Which of the following changes, base substitutions, insertions, or underlined in bold, would have the most severe effect on the protein made from the messenger RNA in the last question? So if we had a change, AUG, AGU with AAG, or UAG, or GUA, number 4 seems to have had seems to make the most, I believe. Let's see. Now we have a deletion there, number 3. So number 3 would be which would have the most severe. That would have the most severe because it would truncate the proper message. So number 3 would be the right answer. Without knowing the translations at all, the third position, as we're going to learn, is the wobble position, and that would have the least impact on the sequence. Most likely, without looking at the table, this would not change the amino acid, but obviously having a deletion certainly would cause that protein not to be functional. Translation is the second step of the two-step process by which DNA directs the synthesis of proteins. In translation, the information from a gene that has been carried by the nucleotide sequence of a messenger RNA is read, and the ingredients present in the cell's cytoplasm are used to produce a protein. So all the things that we've discussed produces and sets up a case for proteins to be made in the proper order. Now, we have any changes in the messenger RNA that results in either deletion or modification can directly affect the performance for the proteins that are made. So this is a problem. So we wanted to point that out. So that finishes section 2 of our presentation, and we'll be going to part 3 next time. So Review this material. Take special care with the diagram showing the overall process of each of these. And I'll talk to you soon.